we are going to attempt to answer questions on cell movement. You will recall that we have discussed cell movement to involve two major types of movement: ciliary movement, in which case the cell or the unicellular organism make use of organelles for movement. Then amoeboid movement is just the flow of cytoplasm within the cell and causing extension of the cell membrane, making this, the organism or the cell to move. That is amoeboid movement. While cyclosis is just the swimming movement of the cytoplasm within the cell. It could be the whole protoplasm moving around a vacuole, or only the cytoplasm with the nucleus in the center flowing around a number of vacuoles in the cell. So we have two types of cyclosis, or we have circulational cyclosis when involve many vacuoles and cytoplasm swimming around them in different directions. Then we have what we call rotational cyclosis, when in this case the protoplasm is swimming around a centrally large vacuole. So with this information, we can now attempt to answer the questions that will involve movement itself. Let's see number one. Which of these organelles is not involved in ciliary movement in cells? So the podium, B, cilium, C, flagellum, D, cilium and flagellum. Take note, organelle that is not involved in ciliary movement. And we define ciliary movement is as movement of the cell in which locomotive organelle is involved. Cilium is a locomotive organelle. Flagellum is a locomotive organelle. The only one that is not locomotive organelle is the podium, which is also referred to as force foot. So A is the answer. Number two. The swimming movement of protoplasm around a central vacuole is called A, rotational cyclosis, B, diffusion, C, circulational cyclosis, and D, endoplasmic reticulum. Then the answer is simply rotational cyclosis, because the whole protoplasm is moving around a centrally positioned vacuole. Number three. Amoeboid movement is possible because of the possession of A, flexible cell wall, B, stationary cytoplasm, C, flexible plasma lemma, and D, flagellum. Now, it's obvious that cell wall is not flexible. It's a rigid structure. D, flagellum is a locomotive organelle. So if it is involved, it should be ciliary movement, not amoeboid movement. If flexibility of plasma lemma will cause movement, that means something must flow into a direction to make the cell membrane change shape and form pseudopodia. So it can never be stationary cytoplasm. The cytoplasm is flowing to make the flexible plasma lemma to change shape. So our answer here is C.